Questions, anybody? I'm not sure why there was a mistake in last week's lab that said you needed to have a broth culture of your unknown, and you made a broth culture, and Friday we didn't use it. We need a broth culture of our unknown for Wednesday because you're going to do a motility test. I would recommend that if your unknown grows pretty well in the incubator and broth overnight, that you do an overnight culture, so you do it tomorrow. I know some of you, though, have unknowns that don't grow particularly well in broth, and if that's the case, you might want to do it today. But we need a fairly young culture to do our motility studies. Wednesday, we're going to do a little bit of medical microbiology. You're going to torture each other and take throat swabs of each other and see what bacteria you have on your tonsils, and do a few skin swabs. So we're going to kind of shift gears right now in class in a number of ways. At the beginning, I really tried to convince you that microorganisms are far more beneficial than they are damaging. But the reality is that there are a number of pathogenic microorganisms that cause serious disease. And so really, for the rest of the semester now, we're going to be talking about disease in one context or another. The first context we're going to be talking about is how do you kill microorganisms that might cause disease? Then we're going to switch over and start talking about some of the adaptations that microorganisms have that make them pathogenic in the first place. And many of those adaptations are actually highly correlated to the symptoms that these particular diseases cause. So once we go through this pathogenesis and disease material, then we're going to switch gears at the end of the semester and talk about what is your body's defense against these particular pathogenic microorganisms. So we're going to be getting into the human immune system and the human response that usually protects you from these things. Occasionally it doesn't, and therefore you could fall prey to one or more microbial diseases. So very kind of disease-related the rest of the semester. And it seems kind of weird to be talking about the rest of the semester, but we're really approaching midway already. Okay, so let's talk about things that kill microorganisms. That was an important part of some of the early learning you did in lab, learning those basic sterile techniques so that you didn't contaminate your unknown, for example, so that you could maintain a pure culture and transfer it to a new growth environment without introducing any additional contamination. I told you last time that this is a pretty good organizing slide in terms of dealing with this information. So we're going to talk about three major categories of strategies used to kill microorganisms, and that is physical methods, mechanical methods, and chemical methods. And associated with a discussion of these various strategies, how they work, what their applications are, what the mechanism of action is some underlying vocabulary that you have to be comfortable with in order to be able to wrap your head around these particular various methods. So, I asked you last time, look over that list of terminology. See if you did it. So what's the difference between sterilization and disinfection? What do we mean when we say something is sterile? Okay, it is free of living life forms, including endospores, because endospores have the capability of giving rise to cellular life forms. What is disinfection? Is disinfection the same as being sterile? No, it isn't. The major difference between something that is disinfected and something that is sterile is that disinfection kills the vegetative cells. Vegetative cells are the ones that just divide and grow. But disinfection does not kill endospores. So that's the major difference between them. Typically, when we talk about disinfection, we're typically talking about chemical treatments primarily. What's the difference between antiseptic and disinfectant? Antiseptics are intended to be used on, or are able to be used on, living tissue, like skin. Disinfectants are designed for use on inanimate objects. So which one is probably more toxic / corrosive? The disinfectants, right? They're harsher chemical treatments, and they would kill skin cells. Typically, antiseptics are also chemical compounds. And so when we talk about chemical destruction of microorganisms, these terms antiseptic and disinfectant come into play. What does the term cidal mean? If you are fratricidal, cidal means to kill, right? What does static mean? Static means to inhibit. And so some agents will actually kill microorganisms and destroy them. And if the agent is removed, the cells are dead and they can't reproduce. Other agents are inhibitory, and as long as the agent's present in the proper concentration, the organisms won't divide. But if you remove the agent, then they're free to start dividing again. So this concept of inhibition versus killing is an important concept. Degermination. Many of you went to Johnson Memorial, and some of you went to surgery, and you saw surgeons very meticulously scrub up prior to surgery, right? That degermination process is a combination of physical and chemical control methods. What's the physical part of degermination? What do they do? Oftentimes brushes, scrub brushes, to remove the skin and any oils adhering to the skin. And then they also use soaps and other materials, which are chemical compounds. And so degermination is a process that involves both physical and chemical treatment of the skin to remove pathogenic microorganisms. How about sanitization? Heard of that one? This substance has been sanitized. Sanitization is a way to reduce the amount of microbes present. It does not sterilize. It does not disinfect, because typically a fairly significant number of microorganisms are still present following sanitization. But it does reduce the numbers. How low? It depends upon what the accepted public health standards are for what you are sanitizing. So over in Saga, for example, dishwashers have to be set at a certain temperature, which have been demonstrated to sanitize your dishes and silverware that you use, reducing the number of microorganisms to an acceptable level, and that acceptable level has been determined to be one that won't make you sick. So either you were very reticent, or most of you didn't look over that material. One of the things that I wanted to give you an alert about is we've got an exam coming up a week from Monday. You should be looking at your study guides, looking at study guide questions. You should be reviewing lecture notes. It's not any too soon to begin some concentrated study.
What was the term for like static is to inhibit?
Cidal is to kill. C-I-D-A-L. And I think all those terms are in that first slide that I showed you that had the terminology associated with controlling microorganisms.
I have a question.
Yeah.
Maybe that's my mistake, but I wrote down that the exam's next Monday.
Yeah.
Oh, I thought you said a week from next.
No, this coming Monday. A week from today. A lot of these particular methods that are used to inhibit or kill microorganisms ultimately are the result of denaturation of molecules in cells, and particularly proteins. Now, some methods have to do with denaturing and destroying DNA molecules, but if you look at it on the whole, most of these physical and chemical methods of destruction basically work ultimately by denaturing proteins. We all know, hopefully, from cell biology and other classes, that both heat and pH changes can cause proteins to denature. And if that changes the shape of the active site of an enzyme, for example, then that can result in metabolic inhibition. And metabolic inhibition can either suspend or slow growth, or it can kill microorganisms. There are also some chemical compounds, particularly heavy metal compounds, which can irreversibly bind to proteins and basically block their active sites. And so this is kind of a chemical form of denaturation. So first we'll talk about heat effects, a little bit later about pH changes, and then tomorrow when we start talking about chemical compounds, we'll come in and talk about some of these chemical modifications of proteins that take place. Killing microorganisms is not as straightforward as it might sound. It's not just a matter of dumping some chemical on them and they're gone. There's a lot of factors that influence how effective these killing agents are. So, for example, temperature has an inhibitory or killing effect on microorganisms. And one of the factors that influences temperature's effectiveness is how long are you exposing the microorganisms to that temperature, and how much heat is there in the case of temperature. So what is the actual temperature, and then how long is the exposure? You can see over here that this particular graph that shows you the log of the viable number of cells versus time, this is in an autoclave. It takes about 15 minutes at 121 degrees Celsius, 15 pounds per square inch. That's the standard autoclaving conditions, for sterilization to occur. And so if you don't autoclave something long enough, there will be a number of viable cells that are left in that particular sample. Usually, to make sure when we autoclave, we go beyond this 15-minute period, and it's fairly standard procedure to autoclave for about 20 minutes just to make sure you've gotten everything. Particularly if the autoclave is really full. So another factor that can influence the effective temperature on killing is the load or the amount of material that you're trying to treat. So you'll see over here two contrasting examples of a partially filled autoclave, one that has a low load and one that has a high load. And you can see that it takes this full 15 minutes for the high load to become sterilized. It takes less time for the low load. Again, we just kind of, by default, say we're going to kind of assume a high load situation because it really doesn't matter if you over-sterilize it in most particular cases. Here's a really important one. Is the organism an endospore producer or not? Because there's a huge difference in the amount of time exposure required to kill organisms that produce endospores versus the non-spore forms. There's also a difference in the temperature required to kill organisms that have endospores. And we'll show you some things in just a second more about that. So these have to do primarily with heat treatment, and in this particular case, autoclaving. Here's an example of factors that are related to chemical treatment to try to kill microorganisms. And in this particular case, they're contrasting what happens when you have a microbiostatic agent versus a microbiocidal agent. In the case of the microbiostatic agent, the cells are still capable of growth if that particular agent is removed. So if it's something that is volatile, for example, and evaporates, then it might only have a temporary effect on inhibition, whereas a truly cidal agent completely kills things. In chemical compounds, chelation is a complicating factor in terms of the effectiveness of chemicals killing microorganisms. What does it mean when something chelates something? Did you talk about chelating agents in organic? Heard that term before? Chelate means to bind to or tie it up. Chelating agents, typically when they bind to chemical compounds, can inactivate them and basically turn them from a cidal agent into a harmless agent. There are a number of chelating agents in soil, in dirt, for example. And that's one of the reasons why you should wash out a cut, for example, that's become contaminated with dirt, not only to remove potential pathogens that might be in the dirt, but to remove chelating agents which could then interfere with other topical treatments you might put on there to try to then kill the microorganisms. So organic matter, dirt and other things, contains a number of chelating agents. Obviously, another thing that is a big determining factor about whether an agent kills effectively or not is what's its concentration. Some chemical compounds at a lower concentration are only inhibitory. They're a static agent. And it's only when you increase the concentration of those agents that they truly become cidal or killing. Now, it probably would seem logical that there's a pretty direct relationship between increasing concentration and toxicity and how effective those agents are in killing microorganisms. But there are a few exceptions to that rule that we'll talk about. For example, if we compared two solutions, one of 100% ethanol and one of 70% ethanol, I think most of us would assume that the 100% ethanol solution would be the more effective inhibitory or killing agent. It's not. Actually, 70% works better than 100% does. Why? We'll talk about that a little bit later on. This illustrates the importance of knowing whether something has endospores or not endospores, what kind of an organism it is. This represents something called the average thermal death time for various cells that could potentially be pathogenic. Thermal death time is the shortest period of exposure at a specific temperature that's required to kill all the pathogens. So what you'll see is that different organisms of different types with different structures have very different thermal death times. And the one that I want to point out particularly here is the difference between spore-forming bacteria and non-spore-forming bacteria. Okay. This shows you a relative comparison of how effective endospores are of preventing microorganisms that have the spores from being killed by a number of methods which are usually quite effective in killing vegetative cells. The methods include moist heat, radiation, in this case X-rays, a sterilizing gas, ethylene oxide, and a sporicidal liquid, a compound called glutaraldehyde. But look at the difference between those that are endospore forming and those that are not endospore forming, and the relative resistance of those that have endospores. 18-fold increase in resistance to these sporicidal liquids if you have endospores. About double for sterilizing gases, about 4 times to X-rays, about 1.5 times to heat. So that's a real issue in terms of killing microorganisms, is knowing whether they're spore producing or not spore producing. Fortunately, a number of pathogens are not endospore producing. There are some pathogenic endospore producers, but they're not that common. Okay, here's something important. There are 2 kinds of heat typically that are used in treatment of microorganisms: moist forms of heat and dry forms of heat. Moist forms of heat are much more efficient at killing than dry heat are, particularly if you compare time of exposure. So look at moist heat. At 121 degrees Celsius, it takes about 15 minutes for sterilization. Dry heat of the same temperature takes 600 minutes. 40-fold difference. Same temperature. Presence of water is the big determining factor. This basically is steam on the top. This is dry heat like an oven. Think about it for a second. If you put your hand in an oven for 5 seconds that's at 212 degrees Fahrenheit, your hand's going to get warm, but you're not going to really damage your hand. But if you take that same hand and put it in a pot of boiling water at 212 degrees, you're going to get serious burns. Why? Because the heat transfer occurs much more rapidly through the liquid environment than it does a gaseous or air environment. The presence of water facilitates denaturation of proteins. Remember, we said that when we talked about endospore structure, and we said that one of the things that makes an endospore resistant is that the core of the endospore is very much like a gel. It's very dehydrated. And under those conditions, proteins don't denature nearly as well. So the presence of that water really facilitates denaturation of the proteins. Basically, dry heat kills by dehydration. Just drying out the cell. And that's why it takes so much longer relative to these methods, which work more on denaturation. When you get up to high enough temperatures, this is a little device you can use instead of a Bunsen burner to sterilize loops. Incineration basically just oxidizes everything. And when you flame your loops, especially if you've gotten quite a bit of growth on that loop, you can see that white ash that forms as you've just fried your microorganisms and just completely oxidized all of the molecules that are present. Well, an autoclave takes advantage of the fact that at increasing pressure, we elevate the temperature, the boiling point of water. At atmospheric pressure, water boils at 100 degrees Celsius. If we increase that to 15 pounds per square inch, the boiling point of water is elevated to 121 degrees Celsius. That extra 21 degrees is critical for being able to kill those resistant endospores. Many people falsely believe that it's the increased pressure that kills the organism. It's not. The increased pressure allows for a larger, higher temperature of water before it boils and turns into steam. The fact that the water is in the steam form within an autoclave helps it to penetrate into the materials that are being sterilized much more readily than dry heat does. So basically what an autoclave is a fancy device that just allows internal pressure to build up in an enclosed chamber and therefore allows water to boil and turn into steam at a higher temperature than it normally would. I didn't show you this, I had intended to. One of the things we're always concerned about is our autoclave working properly? And are there ways to measure whether the autoclave is achieving its desired goal of sterilization? There are a couple of physical methods to do that. One of the things that I throw in there occasionally are some test strips. And these test strips are impregnated with chemicals that are pressure sensitive and change color depending upon the pressure. And so this is a pressure strip with the yellow strip indicating, an untreated strip. And then it turns this kind of olive green if the pressure reaches the proper amount of 15 pounds per square inch. And we're assuming, and it's not always a good assumption, that if the pressure reaches the proper pressure, then the temperature also reaches the proper temperature. Okay? There's some also pressure-sensitive tape that we have, and these little black lines on this tape don't appear until this tape has been exposed to the proper pressure. But the real gold standard for checking how your autoclave is working is using some vials that are shown here that contain strips of endospores of a species of bacteria that is very, very temperature resistant. This particular one is called, you have to remember this name, Bacillus stero-thermophilus. And it can grow happily in an incubator at 50 degrees centigrade. It's a very temperature resistant microorganism. And it also produces endospores. So what we do to check the autoclave using this particular technique is there are these flexible plastic vials, and inside there is this media, and it contains a pH indicator and a fermentable sugar. And then inside this plastic flexible vial is a strip of these endospores. Okay? So what you do is you throw these in the autoclave. And then after autoclaving, you crush the little vial inside by just squeezing on the plastic vial. And that releases the media into the spores. If those spores haven't been killed by the autoclave, then they will germinate. The cells that they produce will start producing sugars. That will lower the pH, and that will change the color of the pH indicator, phenol red, to a yellow color, indicating growth of the microorganisms. If the spores have been killed by the autoclave, then when you crush the vial, the spores are dead, and they can't germinate, and therefore there's nothing to ferment those sugars, and therefore the color of the pH indicator won't change color. So this is a very effective way to biologically test whether the autoclave is working properly. And that's really why we want autoclave, right? We want to kill endospores. And so this is a way to directly show that we're doing that. Unfortunately, these are expensive, and so I typically just check ours at the beginning of the year using this method. Otherwise, I'll just throw in some of these pressure-sensitive things to kind of make sure things are going well throughout the course of the semester. Another technique that involves moist heat, so we've got autoclaving. Is pasteurization. What do you typically associate pasteurization with? Wine? Milk? Yeah, milk was one of the first things that was pasteurized. Pasteur developed the process, obviously, for wine. When I talked about the history of microbiology, I said the French wine industry was really a problem. Because normally what we want to do is we want to have yeast fermenting the sugar in the grapes to produce alcohol. But in the French wine industry, the skins of the grapes were becoming contaminated with the bacterium, and that bacteria would ferment sugar and produce lactic acid, which caused the wine to sour. So what Pasteur deduced was, he said, is there some way to kill the bacteria without killing the yeast? Because we want the yeast to ferment, but we don't want the bacteria to ferment. And he said, well, they have different temperature optimums. And so he devised methods of temperature and time that were designed specifically to kill one microorganism, but not the other. So pasteurization is considered to be a method of disinfection. It is not a matter of sterilization. Boiling something is also considered to be a matter of a method of disinfection, because boiling will kill the vegetative cells, but it will not kill the endospores. So boiling does not sterilize unless it is boiling under pressure, which gives us that additional 21 degrees Celsius temperature. In the pasteurization of milk, there is a pathogen that we are particularly concerned about. And this is a rickettsial disease. I don't know if you remember or not. Rickettsia are those little tiny bacteria we used to think were virus. They're obligate intercellular parasites that can only live inside of other cells. And these particular bacteria cause a milk-borne disease called Q fever. And that is one of the worst and most deadly pathogens that can be transmitted via milk. So pasteurization has been kind of designed with killing this critter in mind. Now, why don't we just boil our milk? Because what's going to happen to the milk? It's going to denature the protein, it's going to curdle. Boiled curdled milk is nasty. Okay. So over time, people have developed, and companies have developed, various methods or types of pasteurization that are distinguished from one another by what is the treatment temperature and what is the time of exposure. So historical batch pasteurization, which was one of the first methods that was used, has a fairly long exposure, but a fairly low temperature. And one of the things that's kind of evolved over time is that new methods of pasteurization, flash, ultra-high, have involved higher temperatures for shorter periods of time, little bursts of intense temperature. There actually is now a method of treatment that's called ultra-high temperature sterilization, so this isn't pasteurization any longer, which actually kills endospores as well, 140 degrees Celsius for 1 to 3 minutes. And if you've ever seen milk that's sold in cartons that's sitting out at room temperature on grocery store shelves, that is ultra-high sterilized milk. They have a lot of that in Europe. Did you, Parker, see milk in the grocery stores that's not refrigerated? Yeah, and it has a little bit different flavor. And if you're not used to drinking it, it tastes kind of odd to you. So one of the things that you've got to be careful about is if you're a company that employs pasteurization, is that you find the conditions that preserves the quality of the food or the beverage that you're treating so it doesn't become unpalatable to consumers. There is another method of moist heat called tyndallization. And there's a device that uses tyndallization called an Arnold sterilizer, and it's not really a sterilizer. I'll tell you why in a second. This particular method exposes substances to free-flowing steam, but that steam is not under pressure. So the temperature of the steam is, about 100 degrees centigrade. And this exposure happens for short periods of time, but over 3 successive days. Over that 3 successive days, you actually can achieve near sterility. So what's the strategy here? The free-flowing steam will kill the vegetative cells, but not kill the endospores. So at the end of day 1, you've got surviving endospores. What are they going to do? They're going to then germinate into vegetative cells. But the next day you hit them again. And that will kill those vegetative cells. And they probably won't have had time in that 1-day period to produce many, if no, endospores. But if they did produce some, then they're going to turn into vegetative cells. On the 3rd day, you knock them out again. There are various serums that are used in tissue culture. Fetal calf serum is a type of serum that we use in tissue culture. And this stuff's got to be sterile. But it's very heat sensitive. And it would denature in an autoclave, and it wouldn't support the growth of the cells in the tissue culture. So one of the first ways that serum was sterilized was using this Arnold sterilizer. Now there are some other more effective methods that are used, and this is kind of falling out of favor. There are some foods that are highly sensitive to temperature that can be treated this way, but there also are other methods now that are replacing this. So this is a summary slide. There are several summary slides scattered throughout these PowerPoints in the next couple of sections that give you nice summaries about the methods and kind of applications, how they work, etc. And so these would be good things to use for review when you're studying this particular material. So these are the methods basically for using heat for sterilization and disinfection. Another physical method that can be quite effective in controlling microbial growth is radiation. Here is our electromagnetic spectrum, aligned with the shorter wavelengths on the left and the longer wavelengths on the right. And you told me last time correctly that there was an inverse relationship between wavelength and energy of these forms of radiation. So the shorter wavelengths have the higher energy, these have the lower energy. And higher energies are basically associated with more lethality. At the highest energies, radiation that's absorbed can actually ionize molecules. They can break bonds. They can cause molecules to polymerize and kind of stick together. And these particular behaviors just play hell with cellular structure. Okay, they really are very effective at destroying cells. Ionizing radiation also produces a number of free radicals. And those free radicals have some of the same outcomes in terms of breaking bonds, oxidizing molecules, etc. So these forms of radiation, including X-rays, gamma rays, are pretty lethal because of their ionizing nature. And we obviously want to prevent exposure to ourselves and our tissues from these particular radiation sources. The ozone layer does a pretty good job of filtering out natural sources of these. We use x-rays obviously in medical settings. And as we discovered more and more, the fact that even low doses and exposures to ionizing radiation can be harmful. Even dental x-rays now, we're concerned about exposure to that level of radiation, and you have a lead-lined apron that you put on when you get your x-rays. Any exposure to man-made sources we try to minimize as much as we possibly can. We'll come back and talk about ionizing radiation in a second with the gamma rays, because there are devices that we can use to generate gamma ray radiation to treat food to prevent its spoilage. That's called irradiation. Slightly longer wavelengths of light include ultraviolet radiation. Still some kick to these, but ultraviolet radiation is considered to be non-ionizing radiation. And it has a different mechanism of killing than the ionizing radiation does. So here's a good example of differences between ionizing and non-ionizing radiation. So here's a radiation source of ionizing radiation. Many physical barriers are very poor at stopping ionizing radiation, and the radiation just passes through them. Lead obviously has an exception to that. It's pretty good at blocking ionizing radiation. But if the cells absorb that ionizing radiation, it can cause damage to key molecules, including DNA. Non-ionizing radiation, if it's absorbed by cells, has a different effect on DNA molecules. It doesn't break those DNA molecules into pieces. It instead causes abnormal bonds to form. And those particular abnormal bonds are called thymine-thymine dimers. Fortunately, because of its longer wavelength and lower energy, non-ionizing radiation is fairly easily blocked by physical barriers. So glass, for example, is even effective in filtering out UV radiation. Water is effective in filtering out UV radiation. Thymine dimer. Thought I had one. Thymine dimers are abnormal bonds. And they form between thymines not on complementary strands. Okay, the bonds that typically hold these ATGC base pairs together are hydrogen bonds. But a thymine-thymine dimer will form between two adjacent thymines on the same strand. And the presence of these dimers interferes with DNA replication and also transcription of DNA and RNA. So they can be pretty damaging. One of the areas where we're particularly interested in foodborne pathogens is in the food industry. There are two types of diseases that can result that are commonly referred to as food poisoning. And technically these are food intoxication and food infection. Most people just lump those two together and call it food poisoning. Okay? What's the difference? In food intoxication, contaminating bacterial cells produce toxins which are released into the person that eats those contaminated foods. These cells don't have to be alive as long as they have previously released their toxin. And it is the toxin that has then effect on certain target organs in the body. We're going to come back and talk about toxins later on, but there are enterotoxins which affect the digestive tract. There are neurotoxins that affect the nervous system. There are hemotoxins which affect the blood. And then they all have some specific pathogenesis. They kill tissues and cause particular diseases. Food infection actually involves ingesting viable cells. And most foods, including meat, poultry, etc., do have some level of contamination of organisms that are on those tissues. And that's why we cook. Cooking is one of the oldest methods, obviously, of trying to kill microorganisms that might contaminate the food that you're eating. And if we don't cook for a long enough time or reach a high enough internal temperature, some of those cells can survive. Remember those organisms that can tolerate higher temperatures temporarily? They're mesophils, but they can tolerate higher temperatures. They're called thermoduric. If they survive, it doesn't take many hours for that population to reestablish itself and for a significant number of cells then to develop and grow in your body and cause various infections in your tissues. So this is a true infection due to cells. This is an intoxication.
Does the pH in the stomach not do anything?
pH in the stomach can kill a lot of organisms, but there are some organisms that can tolerate those pHs and survive. Yep. I mean, really, that's the purpose, one of the big purposes of having that vat of acid sitting in the middle of your body, is that the food that you ingest that is contaminated with microorganisms, most of those are killed by being dropped in that acid vat, but not all. And some of the toxins that are produced are acid stable. So there are a number of major forms of bacterial food poisoning that can come from many different sources. So coming up with good methods for controlling the growth of microorganisms in the food industry is really important. And there are a number of different points in the whole food processing cycle where contaminants can be introduced. And so that makes it even more common. What we'll talk about next time is we'll talk about the use of gamma irradiation in trying to preserve food and prevent outbreaks of food poisoning. So don't forget, unknown either today or tomorrow.